glycolysis. So glycolysis is the first step in aerobic respiration. So what is aerobic respiration? In order to get a strong glimpse of what it is, you have to know one term very well: metabolism. I'm sure this is a term you are all very familiar with. Metabolism just means reactions. So RxN stands for reactions. Reactions where molecules are either broken down or added and synthesized together. So reactions where molecules are synthesized or degraded, broken down. So let's look at each of these terms, okay? The degradation and the synthesis. So when it's degraded, this is known as a catabolic reaction. Okay? So catabolism just means break apart. So it takes the fuel that are found in like macromolecules. I'm sure you're familiar with carbohydrates, fats, and proteins, right? These are macromolecules. They contain energy useful to do work. So this catabolism means it breaks it apart to useful energy. Okay? So takes fuel and breaks apart to make useful energy. And obviously useful is the key term here. Energy that is able to do work, right? So we have a certain amount of force that can be used to push a certain reaction, molecules, atoms, to travel a certain distance or carry energy a certain distance in order to be able to do actual work that the body needs for a chemical reaction to actually proceed. Some products are going to be formed from this. An example, CO2, carbon dioxide, H2O can also be produced. You see how this is looking very similar to the cellular respiration formula, right? Cellular respiration has certain products and it has certain reactants and it produces these products, CO2 and H2O, right? We breathe this out, we sweat this out, for example. On the reactant side over here, that was the glucose, right? Hence why glycolysis is associated with the word glucose, right? Because glucose, it's the most stable hexose, so that just means that it's very efficient to be used as opposed to fats and proteins. So we have glucose here that gets broken up. We also need oxygen, right? So we need glucose and we also need oxygen in order to break them apart and then create these products, okay? So when this occurs, it also generates ATP. So I'll just put here and generates, so I'll just put GN, generates ATP. And ATP pretty much just carries energy around in the cell, okay? So we need energy to actually be creating this molecule here. Once you have that molecule, then for the next reaction, it's going to break apart. That P, right, that phosphate is going to break apart. It's going to release that energy so that continuous chain reactions can occur. So that is catabolism, or the catabolic reaction. Then we have to look at synthesis. Synthesis is obviously the opposite. It produces an anabolic reaction. So let's look over here, anabolic. An anabolic reaction does the opposite. It takes the energy and it uses single precursors, which we're going to be looking at in the next step. It uses those precursors to build complex molecules, so to make bigger molecules, to synthesize building blocks for the body to actually use. So I'm just going to put here, anabolic takes energy and single precursors builds molecules or more complex molecules. Okay, this is all for the body to use. So aerobic respiration is catabolic metabolism. So we're at this step right over here. But it needs oxygen. If oxygen is not present, then it's going to be a very inefficient process. When we look at glycolysis, which is what we're going to be touching upon in this video, it's when oxygen is limited. So let's look at that first step. So again, the different phases are glycolysis, the Krebs cycle, and oxidative phosphorylation. We're looking at glycolysis right now. Just for you to know, oxidative phosphorylation is constituted of two phases. The first phase is going to be our electron transport chain, and the second step within oxidative phosphorylation is ATP synthase. These two, the Krebs cycle and oxidative phosphorylation, will be in another video. We're looking at glycolysis right now, which is the first stage of aerobic respiration. Again, it is not very energy efficient, but it is necessary when you have low amounts of oxygen. So this glycolysis is used in times of stress or demands of exercise where a high intensity, for example, scenarios where you're sprinting or having to do a ton of energy output in a very small amount of time, where lactic acid builds up and you don't have enough oxygen to breathe in sufficiently. So glycolysis, we have to use our glucose that was made in the photosynthesis process from the previous video. So that glucose that was made from one of those G3Ps in photosynthesis gets converted into that glucose, and that glucose is what we're going to be focusing our tenet around here. So we have our glucose. That glucose is going to have to be broken apart. So it's going to break apart to useful energy. Again, in this stage, we don't need actual oxygen. So this glucose is going to be broken up into its constituent parts. What does glucose kind of look like? Well, glucose, it looks like this. There are 6 carbons on the inside, and on the outside we have 2 phosphates. Very important, because we're going to be breaking these apart immediately. So this glucose is going to break apart into 2 3-carbon sugar phosphates. So I'm going to be putting one going up here, one going down here, okay? So if I were to break this in half over there, you see how we have a P with 3 C's, carbons, and another mirrored just below it, because it got cut in half. At this stage, you have to understand that ATPs were broken down. Why? Because it needs to split this glucose apart. This is an exothermic reaction. So it's releasing energy and heat in order for this to occur. Next we have these 3-carbon phosphates, 3-carbon sugar phosphates, having to add an extra P onto this side here and this side over there. So these free-floating P's are going to want to attach onto this. So then we get a P with 3 carbons, and then this one gets attached over there. Same thing with this one over here. It gets attached to this 3-carbon sugar phosphate. So this one now gets onto there at the end. Just for you to note, in order for this to occur, like I said, ATP were used, right? So if you want to put a small disclaimer arrow over there where ATPs were used and were broken apart into ADPs. Okay, and by the way, there's 2 of them, right? So 2 ATPs broke up into 2 ADPs because one of these P's broke apart from this ATP, free floated, attached onto that 3-carbon phosphate, that sugar phosphate, in order to create that one. The other P that got broken off from another ATP started free floating over here, attached onto the end of this 3-carbon sugar phosphate, and then created its next product over there. So now we have this product and this product. In this process, NAD plus is used in order to make NADH. So an electron was transferred in these steps over here. So the NAD plus, I'm going to be putting it right over here. The NAD plus turned into the NADH. This is very, very important because even though we want to be creating ATP, so far we haven't done that. We've actually used ATP. We don't want that. We want to make ATP. Unfortunately, right now we've only been doing the investment phase. We've only been using up ATP in hopes that we can create more ATP later on, kind of like the stock market or an ETF or an index fund or mutual fund, right? You're adding in your current investment of a certain amount of cash so that it can build up later on with potential growth. That's what we're doing over here. So what we've done is this ATP that's been broken apart, we've actually thankfully made NADH. Okay, and we're not going to get into specifics of how that NAD plus turned into the NADH with the help of the ATP. It's a series of many reactions. Just know that for the current selection of this course, you don't need to know the specifics there. Okay, just know that the electrons got floated around thanks to the energy breakup of the ATP in order to turn that NAD plus into that NADH. Okay, again, this happens twice, right? Because this happened up here for these molecules, right? It needs to happen down here as well. So this NAD plus breaks up. So this NAD plus turns into this NADH here as well. So we've created 2 NADH molecules. From there, we need to continue going to the right. So this 3-carbon and 2-phosphate molecule, this sugar, needs to become another product. At this point, we're now going to get just 3 carbons left at the end. How is that possible? It's possible because these phosphates were able to be cleaved off or broken apart, taken off, with the help of what? ADP. All right? And in this case, we had to use 2 ATPs to create the 2 ATPs. So these P's over here were added onto the ADPs, okay? So one of these was added onto one ADP. This was added onto another ADP in order to create that ATP. But what does that leave us with? It leaves us with a molecule here with only 3 carbons, no longer having the P's. Same thing happens over here. We have our 2 ADPs becoming 2 ATPs. Okay, again, this one and this one were used for that process to occur. So this one also has only 3 carbons. This all occurs in the cytoplasm, by the way. So I'm just going to be putting a little star here. Occurs in the cytoplasm. And it's not necessarily a good thing. We want this to occur in the mitochondria. So obviously that's the most optimal place for these chemical reactions to occur. Remember mitochondria in the first video for the organelles, right? So unfortunately this happens in the cytoplasm, not necessarily optimal, but it gets the job done, especially in moments where adrenaline kicks in and you need to produce a high amount of work and effort in a very short amount of time. In times of emergency, it will be perfect. Now, why do we want these 3 carbon molecules? By the way, these are pyruvic acid, or synonymous to pyruvate. We need these because later on they are going to be converted to even more ATP. So right now we've only made 4, right? over here, and over here, but we've also spent 2. So technically you only made a net of 2. 2 ATP net. That's awful. Okay, we went through all this process just to make only 2 ATPs. We need a lot more. So it's going to be converted onto the next steps, our Krebs cycle and our oxidative phosphorylation, in order to be making more of those ATP that is needed. Again, in those scenarios, we need oxygen. So this is why you cannot be doing high intensity exercises for a very long period of time. The intensity is obviously going to decrease up to a certain point because you're just going to get tired. You're not making enough ATP to continue on in that exercise. Let's get into the nitty gritty over here of how this all occurred. So again, we started with our glucose. I'm going to say up here, this is our step 1. All right, so we have our glucose. And we will want to be focusing on glucose because it is the most stable hexose that I said, which means it can easily modify the protein. So it's very important that you have a ample amount of glucose in your diet. Otherwise, building up the proteins, making sure that they fold properly, continues to be ensured. With a diet very low in glucose, this can harm the modification of the proteins, okay, which is obviously not optimal. Glucose is also the only fuel for red blood cells and is the only source for the brain actually under non-starving conditions. So glucose, very, very key over here. We're going to be using that to create our second step. Our second step is when an ATP is used. Okay, so remember that ATP that I talked about in the beginning? So here, maybe I'll break it up here on the side so that you can kind of see what happened. Okay, so I've minimized it here on the side. So we got our glucose turning into glucose 6-phosphate. Glucose 6-phosphate. And that ATP was used. So we'll just say 1 ATP. Okay, so 1 ATP was used in order to create an ADP. Remember, this was the investment phase. So that glucose 6-phosphate, why do we want it to become that? Because in this particular point, we have phosphorylated. We phosphorylated this glucose to have better phosphoryl transfer potential. What does that mean? It's a fancy word of making sure that it stays in the cell. So this glucose is now more anchored within the cell thanks to this process, okay? And now this is a very complex process, using that ATP to turn into ADP using an enzyme, okay? But we're not going to get into the specifics of that. Just know that 1 ATP was used, all right? So times 1. 1 ATP was used in order for that to happen, okay? Now, this happens twice. So you notice how we have 2 ATPs here broken off to turn into 2 ADPs. That's because this one of this 2 ATPs, so 1 out of this 2 ATP was used in this particular direction for one of those 3 carbon sugar phosphates, okay? The other ATP from this one was in this direction for the other sugar molecule, okay? So I know it says times 1 over here. It's because this whole process that I'm going to be exemplifying for you right now happens twice, okay? So this is only going to be one step, okay? This is going to be mirrored again, okay? But obviously we don't want to be drawing 2 of them happening at the same time. It's kind of a waste of space and a waste of energy, okay, for me to show you. Just know that we're just showing you the first step from this glucose turning into that pyruvate over there, and then just know that this repeats again, okay? So that 1 ATP from that glucose turned into the glucose 6-phosphate. Now we eventually get to our next phase, okay? So this is still going to be part of step 2, okay? But this becomes now fructose 6-phosphate. Why do we want this to occur? To make this readily cleavable, okay? So it's readily cleavable this way. So we have our fructose 6-phosphate. It's pretty much the same as this glucose 6-phosphate. It's just an isomer. That just means it's the exact same structure, same bonds, same atoms. Okay, but they're just the bonds are rearranged in a different format. Okay, but it's still got the same constituent materials. So we have our fructose 6-phosphate now. Now it's in a more readily cleavable state. Now we're going to get to our step 3. At this point, we've now used another ATP. Okay, so our ATP was used again here, so I'll just show it to you like that. Our ATP to ADP. To turn it into fructose 1,6-biphosphate. So what happened here? It got phosphorylated again. All right, remember it happened over here, right? So this was our first phosphorylation. This here is our second phosphorylation. Remember, it's to make sure that it stays within the cell. Okay, so that happened to make sure it stays within the cell. This one also to make sure that it stays within the cell. So the ATP over here, one of its P's got added on to this glucose. So this glucose now has one of these P's. At this point, our fructose 6-phosphate, because it looks very, very similar to our glucose 6-phosphate, it still only has one P. Because we did another ATP to be broken up, to be invested, we took that P from that ATP, added it on to this fructose 6-phosphate, so that now it is a biphosphate. It has 2 phosphates right now. Okay? So now we have 2 phosphates. That's why you can see here in our little drawing on the side, you see how it started with just, these got cleaved off right in the beginning. But then eventually this 1, 2, 3, 4, 5, 6 carbon, okay, split off in half. But then that 1P came from this phosphorylation. Okay, so now there's 1P over there. Guess what? It happens again, right? That ATP gets broken up into ADP. One of its P's got added onto that fructose 6-phosphate, and that's what we have over here, okay? So it got added onto here. So now this thing over there is a fructose 1,6 biphosphate, okay? And as you can see later on, we're going to be losing those, but we're not there yet. We're still on step 3, okay? So we have that fructose 1,6 biphosphate right now, again with the help of an enzyme, okay? A different enzyme actually. But anyways, let's go to step 4, okay? So let's go to step 4. That fructose 1,6 biphosphate turns into a GAP. This GAP is actually ready for the next step in glycolysis, okay? So right now, our steps 1 to 4, okay, is actually the first stage of glycolysis. We got to get to our next stage, okay? So this GAP is actually ready, okay? So this GAP is ready to go from here in order, from here to there, okay? So technically all of this that's happened, okay, is just the first stage, okay? The second stage is going from here to here. In this case, our GAP molecule, it is ready. But unfortunately, this fructose 1,6 biphosphate creates a DHAP molecule. And this DHAP molecule is unfortunately not ready. It needs to go through extra steps in order to turn into that GAP, okay? So again, we're not going to get into the nicks and crannies of DHAP with its series of reactions to turn into GAP. Just know that it requires extra steps with the help of certain precursors to turn into another GAP, okay? But eventually it does happen, okay? So now it turns into a GAP, now it's ready, and now we have this GAP over here ready to become its pyruvate, and then we have this GAP over here within its reactions to turn into its also another pyruvate as well. So all of this was actually steps 1 to 4, but to go from DHAP to GAP, it's actually a fifth step. So we've done steps 1 to 5 from here all the way to about here. Okay, so now we're just going to go from here to finishing it off. Okay, our second step. So our second phase. Okay, so our first phase we went steps 1 to 5. Now we'll go from steps 5 to I think 10. Okay, to make that pyruvate. Okay, so a lot of steps happen. Okay, within that particular area over here. So let's start getting our GAPs. Okay, we're just going to be doing GAPs just once, and then we're just going to duplicate it. All right, so we're at our fifth step over here with our GAP molecule. Remember that this happens twice, okay? It happens simultaneously. So we're just looking at one GAP over here becoming that pyruvate. So I'm just going to show it to you once here on the side. Just know that it happens simultaneously at the same time again, okay? So we're going to be getting 2 of these GAPs turning into the pyruvates. Okay, I'm just showing it once over here for simplicity purposes. So our GAP, our 5th step is going to become next our 1,3-biphosphoglycerate. Okay, just for simplicity, we'll say 1,3-BPG. Okay, so 1,3-BPG. In order for that to occur, a high phosphorotransfer potential gets added. Okay, so this NADH gets produced. In order to get to our next step, for step number 7, it turns into 3-phosphoglycerate. What does that mean? That means one of these P's got removed. How? With the help of a release of energy using ATP. Okay, so that ADP now turns into ATP by taking one of these P's, removing it, and adding it onto itself, the ADP, so that it becomes ATP. So now instead of biphospho, we only have one phospho. So we only have one phosphorus right now, okay? So that molecule that we had here initially, we removed one of those P's, okay? As you can see happening up there, right? One of those P's got added onto the ADP. Now it became an ATP. And it's going to obviously happen again, right? Because obviously you see 2 P's over there. We have none over here. So that would be steps 8, 9, and 10, right? So we've only removed one. We got to remove another one, okay? So we have 3-phosphoglycerate right now. I'll just say at this point there was a release of energy. So a release of energy. Now we got to keep going, okay? So now we got to get to step 8. So step 8, it turns into 2-phosphoglycerate, okay? So at this point, it's not removing another P. It's just adding, at this point, another phosphorotransfer potential, okay? So adds another phosphorotransfer potential. Next, we have step 9. This creates phosphoenol pyruvate. This is an extremely unstable position. Okay, so I'll just put here as a star, very, very unstable. But it's actually a good thing that it's unstable because it's jam-packed together with certain bonds, just waiting, just absolutely waiting to be released. Okay, it's kind of like when you're on a diving board, you're jumping on the diving board. The diving board is bent, right? It's bent extremely to a very high degree, and it's just waiting for that stress to be alleviated, okay? And then as soon as you jump off that diving board, it releases a ton of that potential, okay? That potential energy that's just growing and growing and growing becomes that kinetic energy and springs you off so that you can jump into the water. right? Or dive into the water. So that stable, that unstable format over here is needed so that we can go on to step 10. All right, so step 10 turns into pyruvate. And it's just waiting, it's eager for this step to occur, right? From step 9 to step 10. So that ADP, again, at this point, turns into ATP by taking off one of those P's over here. Okay, so one of those P's gets added on to the ADP to turn into the ATP. So now you have a pyruvate with all of the P's now removed. Okay, hence why you have that. Okay, just a 3-carbon molecule. Okay, so that would be our pyruvate. Remember, this happens twice. Okay, so I just put a big ×2 over here. This happens a second time, happening simultaneously. So now we have 2 pyruvates made. Okay, we got a pyruvate made there, and we've got another pyruvate made there, and we've made 2 ATPs, right? So we've had an ATP over there, added another ATP over there. So we've made 2 ATPs, but we have 2 ATPs times 2 because it happens again down here as well. So we've actually made 4 ATPs. The problem is we've actually spent 2 ATPs before that split off into 2 things happening at the same time, right? So we actually used 2 ATPs at the very beginning. So it's 4 minus 2 gives you 2, so we actually made a net of 2, okay? So that's why I made the disclaimer in the very beginning. We only made 2 ATPs, okay? But in terms of the NADHs, we've made 1 over here, 1 over there, okay? We didn't invest any, we didn't use up any in the beginning here, so we actually have a net of 2 NADHs as well. I also want to quickly preface it that our oxygens need to be present to continue in this process. Okay, so if oxygen is present, we can get our pyruvate. Remember our molecule that kind of looks like that, our pyruvate, to turn into acetyl-CoA, necessary for the second step, right? Krebs cycle of the step of aerobic respiration. So in order for it to get to the Krebs cycle, it requires sufficient amount of oxygen. Okay, so this will be the Krebs cycle. If, however, oxygen is not present, so let's put this down here. If oxygen not present, that pyruvate, right? That pyruvate cannot become acetyl-CoA. It actually undergoes fermentation, okay? Also known as the anaerobic respiration category, okay? We do not want to convert this pyruvate that we so desperately made to turn it into this acetyl-CoA if there is no oxygen, because we can't use it. And as soon as you get pyruvate to turn into acetyl-CoA, it's actually an irreversible reaction. It's very energy taxing, okay? So it's going to be a nightmare for this to turn into this if oxygen is not present to continue the steps following it. So we just don't even want to go down that route. So we would just go from the pyruvate into fermentation, okay? And it's obviously a very inefficient process. We don't want this to occur, but in certain times of emergency, we would have to, okay? But we're just going to mostly focus on the next video in the Krebs cycle, that oxygen is plentiful and is sufficient in number so that we can just go into that Krebs cycle, eventually go on to the third step of aerobic respiration, which is oxidative phosphorylation, and then continue to make more and more ATP.